Hello and welcome to BioRules video lessons. Today we are going to be talking about cell divisions and how cell division is related to genes, chromosomes and genetics. So, first thing, every cell comes from different cells. This is cell theory that I saw before, and the cell has a cycle. Some cells, they grow after they have been divided and they don't do anything more. They keep doing their life, they don't divide anymore. But some cells, they are going to go through cell division, through mitosis, sometimes through meiosis. Okay? So the idea here, in this beginning of the class, we're going to see the preparation for cell division. So before cell division, the cell goes through a preparation for cell division, either for mitosis or for meiosis. So the idea is that the cell is going to be duplicating the genetic material to be able to make two cells with that cell material. So the cell cycle, the overall cell cycle, is going to be something like this. The cell starts by having shown up from a division from another cell. And so in that moment on, there will be something that we call interphase. Interphase in between the phases of the mitosis or meiosis. This interphase is the moment that the cell grows after being divided, and also when the cell gets prepared if it's going to be dividing again. So the first phase is called G1 or growth 1. That one will happen no matter the type of cell that we have, if they're going to be dividing more later or not. A cell, just after it has been formed by division, it's small and needs to grow. So the cell grows, the cell builds more organelles, the cell goes back from being half a cell to being the size of a regular cell of its kind. When the cell is going to be dividing, when now comes to the moment in the life of a cell that it thinks, oh, maybe I should become two cells, maybe I should divide, then this is going to grow from the growth phase into the S phase, also known as the synthesis phase or DNA replication phase. In this moment, the DNA is replicated, DNA is copied. And when you had one set of DNA, now you're going to have two sets of DNA. So you have twice as much DNA. So the cell after synthesis is going to be with the genetic material ready to be divided in half. After synthesis, if the cell is going to be dividing, it came to the moment in the life of the cell, you say, oh, I want to divide, there will come another growth moment that the cell grows a little bit more, sometimes make a little bit more organelles, preparing for the cell division. And this is interphase. When the cell decides to go through division, then it's going to go through the stage that we call M, which is going to be mitosis or meiosis. So this is going to be mitosis, meiosis, depending on the cell division that we are talking about. So this class, we are going to see here the interphase, and we're going to be seeing the typical cell division, which is going to be mitosis. So what happens in the beginning of the interphase? This is going to be representing our cell. It has genetic material inside. I made here 4 chromosomes, so 4 tapes of chromosomes. But it's too messy over there, so I made a representation here on the right. We have a green and a yellow that is slightly smaller than the blue and the pink. So the green and yellow are homologous chromosomes or a pair, and the pink and the blue, they are another homologous chromosome or another pair. So this one here outside, it doesn't exist. This here is just to illustrate what's going on in here in the nucleus of the cell. So in the beginning of the interphase, what's going to be happening? We have that cell that's in G1, so in G1 it's just grew. And then comes synthesis. In synthesis, what's going to happen, if I even slow down here, for you to see what's going on there in the middle, oh look, whatever was one tape of DNA now became an X, which is 2 tapes of DNA or 2 chromatids. So this is going to be quite important. At this moment here, this cell has 4 chromosomes, each chromosome with 1 chromatid. And at this moment here, the cell still has 4 chromosomes, but now each chromosome contains 2 chromatids because replication just happened. From this moment on, the cell is going to be growing, so it grows again in G2, and the chromosomes stay the same, it's just a little bit bigger, maybe with a little bit more organelles. This here is interphase going on in front of us. It starts with a certain number of chromosomes, it ends with that number of chromosomes, but each chromosome now has twice as many chromatids. Okay? So, now let's talk about cell division. So, during mitosis, the cell is going to be dividing, and it's going to be going through a series of phases or a series of steps where some stuff are going to be happening for one cell to become two cells. So the first phase, which is called prophase, is the initial one. We start with the end of interphase, what you have here. The chromosomes are with twice as many chromatids, so you have two chromatids in each. This cell is ready for division. And then prophase goes on. This here is going to be prophase. The first thing that is visible during prophase is that the chromosomes that were all loose here in the middle, they become super coiled. They coil like this, making a chromosome and like that. So this here is super coiling. So what you have here, chromosomes are super coiled. What's going to be happening next? The nuclear envelope will disappear, will fade. So it's broken down. So nuclear envelope disappears or breaks down. Also, the centrioles that were there very close to the nucleus, they migrate to opposite poles of the cell. In animals have centrioles. They're going to be doing this to help the spindles to be built and organized. And after the centrioles migrate to the poles, what's going to be happening? Spindles are formed. Microtubes are formed, and they attach from the centriole to the middle of the chromosome, to the centromere, that place where the two axes, the two sides of the X, where they meet. So basically they're going to be connected in here. So each chromosome now is going to have its own spindle, its own microfiber. So spindles are formed and attached to the centromere. So this is what's going to happen during prophase. Lots of stuff. After prophase, so let's start here, putting the prophase again, that's a cell with prophase. We have another stage called metaphase. Metaphase, a very important one. What's going to happen now is that those microtubes, they are going to be shortened or elongated and moved, and they are going to be pulling the chromosomes to the center of the cell, around the equator, but in the center of the cell. So basically, if this is a round cell, they are going to be something like that all over the surface, very close to the surface of the cell, around the equator, and they are getting organized there in the middle. So metaphase, chromosomes get aligned in the cell's equator. That's metaphase. That happens in mitosis. They are there one by one, aligned in the middle. That's it. Okay. Next stage. Starting with metaphase, we have the next step, which is going to be anaphase. So anaphase is also quite simple, not very complicated. The spindles that are not connected to chromosomes, they elongate, they get longer. So the centrioles move a little bit away from each other. And the spindles that are connected to chromosomes, they shorten. And when they shorten, they separate the sister chromatids. They pull the sister chromatids apart, they break down that X, they separate them. At this very moment, this cell contains twice as many chromosomes. So if it had 4 chromosomes there, now it has 8 chromosomes for a short amount of time, because the chromosomes have just been broken apart, and now each chromosome has 1 chromatid again, and they are moving to opposite poles. So attached spindle shorten, the sister chromatids, they are separated and they move to the opposite sides, the opposite poles of the cell. This is anaphase. Ana separates. Okay, and finally, if you have here anaphase in there with us, what's going to be happening is that during a stage called telophase, what's going to happen? Look. The nuclear envelope is going to be rebuilt. So the nuclear envelope shows up again. The cell division is almost over. The chromosomes that were supercoiled, they lose a little bit, so they uncoil. Okay, and we have something called cytokinesis, which is that the cell strangles itself in animals, and they separate into cells. So they break down the cell. So half the organelles go from to one side, half goes to the other side. The cell now is divided, mitosis is done. Okay, and as a result, what we have, 4 chromosomes in here, exactly what we had in the beginning. 4 in one side, one cell, 4 in the other cell. This is mitosis, starting with a certain number of chromosomes, finishing with the same number of chromosomes. Mitosis. Okay, putting everything together just to make sure that you are analyzing everything here. We have G1, it grows. Then we have the synthesis phase where the DNA is replicated. The chromosomes now have 2 chromatids each. Then it grows again, G2. There it goes. And then we're going to start mitosis. So we have here the prophase, nuclear envelope breaking down, supercoil of chromosomes, spindle being formed, centrioles migrating to the poles, and then in metaphase the chromosomes are aligned in the equator of the cell. And then you have anaphase where the sister chromatids are separated, they are pulled apart to the opposite poles, and finally we have the formation of the nuclear envelope uncoiling the chromosomes and cytokinesis breaking down the cell in 2. And what we have here, 2 cells exactly identical to the original one. The original one had 4 chromosomes, a green, a yellow, a blue, and a pink. The 2 new cells have 4 chromosomes, pink, yellow, blue, and green. So exactly the same. This is mitosis. Okay, so putting everything together, mitosis is going to be cell division. It keeps the number of chromosomes. It's not going to be change diploid, the number of chromosomes of a cell. Okay, interphase always replicate chromosomes before mitosis, so interphase is not part of mitosis. Okay, it doubles the chromatids but keeps the number of chromosomes, and you have the separation of the sister chromatids in mitosis. This here is the process of mitosis. In plant cells, things are slightly different. Okay, what really matters with the genetic material is not different. Okay, you have the spindles being formed, attached to the centromeres of the chromosomes, exactly the same. The difference is that there are no centrioles. So the spindles are not so organized into one single point like you have in animals when there are centrioles. That's one difference between plants and animals. Okay, there are no centrioles, and the spindles get a little bit more diffused. Another difference between animals and plants, that plants during cytokinesis you don't have a strangling of the cell because the cell have a cell wall. It's hard. So instead of strangling to be able to separate the 2 cells apart, a disc will be formed there in the middle, a plate that is built with cell wall, and then the cytoplasm follows that and separates the 2 cells. So we have a central disc or a central plate that's made, allowing that the cell walls break into 2 cells and the cytoplasm, and you have the cytokinesis. This is the difference between plants and animals. Okay? This cell cycle is controlled by chemicals, as practically everything in organisms' bodies. They are controlled by chemicals, they are controlled by signalers, they are controlled by enzymes, hormones, whatever. In the case of cell division, they are controlled by cyclins. So cyclins are chemicals that, depending on the concentration, they are going to be triggering something on the cell cycle to happen. Okay? So we have here, for example, cyclin D that grows at the beginning of the cell cycle and then ends up dropping in the end. We have another one here, the cyclin E, that goes up more or less in the beginning, but not so much in the beginning as the cyclin D, and drops almost immediately after. We have another one here, cyclin A, that increases after the E and then drops before the end. We have here cyclin B that increases almost there in the very end, and it drops also there in the end. So what's happening here? Let's mark. This is the G1, G2 phase, and then you have mitosis over there. So if we have to say here, what's responsible for controlling the overall cell division? Cyclin D. It grows, it's going to be regulating probably the other cyclins. Cyclin E signals for you to start the S phase, synthesis phase. Cyclin A it starts showing up in the S phase, but it peaks during the G2 phase. So it probably signals finishing the S phase and beginning and doing the G2 phase. And cyclin B, very, very important. That one triggers say, okay, we are about to start mitosis. Let's divide the cell in two cells identical to the original one. Those are the cyclins and how they work. Okay, I hope you had fun. There is something a little bit more, but I'll show this in class, related to mitotic index. How can we quantify the amount of mitosis is happening with cells? But you're going to be seeing this in class. Maybe you're going to see that in your book. Keep your eyes open for mitotic index. Okay, I see you in class. Bye bye.